What if we already have alien disclosure? What if I told you you can learn what aliens are made of, how they function, and even what they believe? In 2023, a highly detailed anonymous post surfaced on Reddit, claiming to be a leak from a molecular biologist. For years, this scientist allegedly worked under a classified national security contract to map the genetic blueprint of what the government calls EBOs, Exo Biospheric Organisms, what we would call the Greys. Before the post was abruptly scrubbed from the internet, it was highly debated. I want to go through this massive claim, attempt to translate this person's complex jargon into plain English, and then make connections to the overall UFO and alien lore and beyond. Is this real? Stick around and let's try and figure it out together.
In fact, all the indications are the aliens eliminated microbial and viral life from their own ecosystem long ago.
Full disclosure from me, haha. Initially, I was going to cover the infamous 4chan underwater UFO base in this video. But the deeper I dive into that supposed leak, the more interesting connections and sources I find. That might be my longest video yet. Stay tuned as I will definitely cover that supposed leak in the future. But now let's begin our deep dive into what we will call the EBO leaker. For context, I want to discuss the odd way this post was completely wiped on Reddit. The leaker posted this highly detailed post, then was answering questions on Reddit. About 5 hours later, as we can see here from multiple moderators of the alien subreddit, the user's account was shadow banned by Reddit itself, which means that they couldn't comment or post. Not long after, the post itself was deleted, along with the user's account. Luckily, there are archives of the post. Keep this in mind as we start to dig in. Admittedly, I'm not a biologist. I will do my best to explain and understand the technical side. Giving explanations of acronyms or jargon should be easy enough at least. Let's break it down paragraph by paragraph. From the late 2000s to the mid 2010s, I worked as a molecular biologist for a national security contractor in a program to study Exo Biospheric Organisms, or EBOs. The aim of the program was to elucidate or map the genome and proteome, which are the proteins, of these organisms. Although the study of OBCs, or other biological constructs, has been going on for decades in other programs, the new high-throughput DNA sequencing technologies of the late 90s unblocked stagnant research in this area. Since then, several breakthroughs have led to significant advantages in our understanding of the genome and proteome of these beings. What we've learned so far has enabled us to outline some disconcerting perspectives about our place in the universe. Briefly, we've discovered that the EBO genome is a chimera of genomes from our biosphere and from an unknown one. They are artificial, ephemeral, and disposable organisms created for a purpose that partially eludes us. I'll be substantiating my statements after a brief introduction. It appears the so-called Greys are manufactured drones, or at least gene-edited, created with multiple different genomes combined, part from Earth, part from somewhere else.
I think that part of what we're seeing here, I mean, look, if you're an intelligence, are you going to go down on a planet with a bunch of angry monkeys who might kill you? No, unlikely. You'll send some intermediary. Well, what kind of intermediary are you going to send? You're going to send something that maybe almost looks like them, but isn't them. So I think, and this is, again, from inside the intelligence community, most of what we think we're seeing are avatars, biological robots that are basically put there to be the minions, if you will.
We've heard that in connection to supposed aliens before, plenty of times. However, this leak gets a lot more technical below. I do want to take a guess at the supposed technology that helped with breakthroughs that was developed in the late 90s mentioned by the leaker. In my opinion, the leaker is likely referring to the ABI Prism 3700 DNA analyzer, which is a mouthful. It was developed in 1998 by PE Biosystems. According to research, it was the first sequencer that allowed for reading of DNA unattended 24 hours a day, which would help with the Human Genome Project. Moving on with the leaker, the reason for disclosing these secrets is quite simple. I believe that every human being has the right to know the truth, and that to progress, humanity needs to divest itself of certain institutions and organizations that will probably not survive these revelations in the long term. I'm aware that I'll have very little impact in this regard, but I still believe that small leaks are necessary to break the dam of misinformation on this subject. When the governments will eventually reveal those secrets, there will undoubtedly be a societal upheaval, but in my opinion, the longer we wait, the worse it will be. I choose to divulge what I know anonymously out of selfishness for the well-being of my family and myself. I'm aware that this diminishes the reach of my credibility and my message, but it's the furthest that I'm willing to go. I choose this forum because it offers a good compromise between anonymity and popularity. In order to protect my anonymity, I will be purposefully vague or even contradictory about any information that could identify me. I'll even introduce red herrings in this respect. I want to make it clear that any information related to the subject of the research will not be treated this way. I'm gonna guess some of the institutions that are referenced here are probably global financial, religious, and even political institutions.
And certainly to execute a plan disclosing this information, there's certainly theological, socioeconomic, other implications. In the news, you're seeing a lot of talk about demons and other things like that. You know, the Vice President Vance has mentioned that, but I think it's theologically premature without knowing everything, equating everything to demons.
Also remember, the EBO leaker mentions potential red herrings, but only with talk of their background, not with the science behind the EBOs. Further down, when we talk about their workplace and their background, keep this in mind. From the leaker: before going any further, please excuse me if you find it difficult to understand what I'm explaining. Some parts of my text are very technical. It's difficult to find the right balance between vulgarization and scientific explanation. I'll continue by talking about myself. What's the point of talking about me, knowing that the information will necessarily be misleading? I simply want to introduce a perspective on the type of people who work there, normal scientists. I have a PhD in molecular biology. I didn't actively seek to be part of this program. Rather, it was a stroke of luck that introduced me to one of the senior scientists. I met this person at a conference when I was presenting a poster on my PhD research. When I think back, I don't believe he was impressed by what I was presenting, because it was quite frankly a project that wasn't going anywhere. I think it was rather the most important aspect of professional life, the attitude and ease with which you make connections. Shortly afterwards, I graduated and received a call from this person offering me a position. At the time, everything pointed to me working in a regular laboratory. Remember, they mentioned red herrings? I'm questioning everything about their meeting and how they got onto the project. You would think that they'd have to be impressed by their science to offer them a position. Quote, I did a series of 3 increasingly suspicious interviews, each at a different location, where my scientific background and knowledge became less and less relevant. The first was with 2 of the senior scientists. The second and third with people I've never seen again and who were obviously not interested in science. Sometime after the interview, I was asked to go to a 4th location where what seemed like a corporate lawyer presented to me with an NDA. He made sure to not only explain every detail, but also that I understood the consequences of not respecting it. The first employment weeks were by far the most memorable, although I spent most of that time in a depressing archive room. It consists almost exclusively of reading about the subject of study and to get us up to speed. There's no secret Wikipedia or even a reference book to guide us. There are only dry reports, memos, presentations, procedures, and SOPs, or standard operating procedures. These documents are almost exclusively about the biology of EBOs, but there are also a few that deal with other subjects such as their food, religion, or culture. There are no documents on their technology. The supposed leaker's initial interview process, though vague, reminds me of whistleblower Bob Lazar's supposed interview process.
And he gave me a contact at EG&G Special Projects. So I went down there. They were, at that time, they were based at McCarran Airport in Las Vegas. Yep. Big building.
The questions more scientifically oriented, or were they personal?
No, the second set of questions were almost exclusively what I did in my spare time. The first one was
like
all
hobbies?
Technical stuff. Yeah. What do you do? What do you do when you leave work? What do you do here? What have you built anything? It was all after the fact question.
As part of my indoctrination into the program at S4, I would randomly be taken into a small room which contained a table, a chair, and 120 or so briefings in blue folders. I'd be left there to read for varying amounts of time, usually about half an hour. These briefings contained a wide spectrum of information, mostly relating to aliens and alien technology. These reports appeared to be an overview of alien information which can be used to brief scientists from any field about the scope of the whole project, and not just their specific field of endeavor. The overview of Project Galileo was accurate. I read the overview and later witnessed evidence which proved it to be accurate. So it is possible that scientists involved with other projects could have seen evidence that these other overviews were also accurate, but I can't make that assertion.
Multiple interviews at locations, briefings of documents about the topic, and even extra about their culture. As mentioned above, the aim of the project is to gain a better understanding of the EBO genome and proteome. To achieve this, a team of around 20 scientists, 4 senior scientists, and a director was involved. Scientists, like myself, had their main responsibility to carry out the technical work. As each scientist had, to my knowledge, a PhD, we were all somewhat overqualified for what is ultimately a technician's job. As senior scientists who make full use of their diplomas had the task of designing the assays and a supervisory responsibility. They were also in charge of training new employees, and sometimes even came in to do technical work. The director, of course, was the person in charge who dictated priorities to the senior scientist. He was rarely on site, and the few times he was, it was to attend meetings. Other than scientific staff, there were security guards working for one subcontractor or another. There were no support staff, such as janitors or maintenance workers. Scientists were responsible for this kind of work. In addition, logistics constraints assured that every scientist is capable of carrying out any technical activity. This paragraph is pretty self-explanatory. Less people, in theory, means less leaks, although we maybe have one here. The next paragraph is a big one. The laboratory itself is located in Fort Detrick, Maryland, in a building used for legitimate biomedical research. The clandestine operations are carried out in a restricted part of the basement, out of sight from regular workers. Contrary to what one might imagine, the biosafety level is not maximal for this type of research. Indeed, the lab containing EBO samples, or derived cell cultures, is BSL-3, while the lab where assays were conducted was only BSL-2. The BSL-3 area of the facility includes a freezer room and a cell culture lab, and is only accessible through an antechamber from the BSL-2 section. EBO carcasses are preserved in horizontal freezers at a temperature of minus 80 degrees Celsius nominal. To maximize the preservation of these carcasses, they are preserved in vacuum bags, and the air in the room is controlled to minimize humidity. There are only 4 bodies, and none of them are complete. It's obvious that these creatures have died as a result of major trauma. I've never witnessed a motorcycle accident fatality, but it probably looks similar to this. It is acknowledged that there are EBO carcasses at other locations. The cell culture laboratory, as its name suggests, is where the cell lines derived from EBOs are grown and related activities are performed. I'll talk in more detail about these specific cell lines later on. The BSL-2 part is mainly used for assays or tests, immunohistochemistry, which is checking which proteins are present, genetic engineering, immunocytochemistry, which is staining cells to study them, and storage, etc. There's also a cell culture lab, but this is used for more traditional cell lines. Other than the labs, there are all amenities you could find in an office. Note that the internet access is limited to senior staff and up. There is, however, an intranet for bioinformatics needs. Okay, let's talk about where this leaker supposedly worked. They name dropped Fort Detrick, Maryland. Later, in a comment that was deleted, they revealed they worked at Battelle National Biodefense Institute. Now this could be a red herring. However, as we see here, this is what a BSL-3 lab looks like in this facility. This video came out after the leak. The lab focuses on biodefense and biological threat analysis. It does seem like a legitimate place where this kind of research would happen. We continue. On the subject of the biology of these beings, I'll start by discussing genetics, then their gross anatomy, and finally their biological systems. For the sake of clarity, the information that I provide here is an aggregation of what I have observed and what I have read. I will make many comparisons with human anatomy because it is the most logical reference. Just a note from me here, sorry in advance if I butcher pronunciation of any of these technical words or aspects. Any biologists listening, please comment down below your thoughts. and that goes for anybody else as well. First, I'd like to discuss their genetics. Their genetics are like ours, based on DNA. This fact was very puzzling for me when I first learned about it. We imagine that beings from an alternate biosphere would have genetics based on a completely foreign biochemical system, and surprisingly, this is not the case. Several conclusions can be drawn from this surprising revelation. The one that immediately comes to mind is that our biosphere and theirs share a common ancestry. They're eukaryotes, which means complex life forms whose cells contain nuclei, like humans, animals, and plants, which suggests that their biosphere would have been separated from ours sometime after the appearance of this type of organism. The term Exo Biospheric Organism is actually a misnomer, which is a technically wrong term, but as it's a historical term, it's still used. Their genetics are not only based on the same genetic system, but they're also even compatible with our own cellular machinery. This means that you can take a human gene and insert it into an EBO cell, and that the gene will be translated into protein. And this of course works in reverse with a human gene inserted into an EBO cell. There are important differences in post-translational modifications that will make the final protein non-functional, but I'll discuss those later. Their genome consists of 16 circular chromosomes, which humans have 46. The term EBO itself is interesting. There are plenty of references to the term EBEs, or extraterrestrial biological entity.
Well, the whole thing started in July 2nd, 1947. That was the first crash.
First crash of an alien spaceship.
Of an alien spaceship.
We recovered alien bodies from that incident. Right?
4 from that particular crash, yes.
And what'd they do with them?
They autopsied them. Dr. Detlef Bronk was the chief surgeon, so to speak. He was the one that named them EBEs. It stands for extraterrestrial biological entities.
Not exactly the same, and you wonder if one of the terms is misinformation. Also, with their genetics being comparable to humanity's, and even proteins being accepted both ways, this obviously lends the long-standing rumors of operations being done on humans, with the end result potentially being some sort of hybridization. That claim has a long time been a part of UFO and alien lore. Again, as we will continue to see, this is a highly technical, sounding at least, leak that weaves in and out of common UFO lore, and we are trying to make connections where we can. Quote, "You're probably familiar with the concept of the inner genetic region, or junk DNA. They are basically DNA sequences that don't code for proteins. These are evolutionary residues. Transposons, or DNA sequences that can change their position within a genome, inactivated genes, genes that have lost their function through mutations, and so on. To give you an idea, in humans, inner genetic regions represent approximately 99% of our genome. I'm aware that these sequences aren't completely useless. They can be used as histone anchors, as buffers to protect coding DNA from radiation, or even as alternative opening reading frames, but that's rather peripheral. What's particularly striking about the EBO genome is the uniformity with these inner genetic regions. We see the same sequences repeated everywhere, and the distance in BP, or base pairs, between the genes is virtually the same throughout their genome. The result is a minimalist, highly condensed genome. In fact, it's much smaller than ours. Moreover, the quality of protein coding genes is even significantly lower than ours, probably due to the genetic refinement, but also due to biological processes that are absent in an EBO. The uniformity of these sequences is a major indication of the artificiality of these beings. There is no complex organism on Earth that has such elegance in its sequences. There is no evolutionary pressure that can lead to this kind of characteristic other than genetic engineering. The EBO leaker is stating that these beings are engineered to reach the level of complexity they are designed to not be wasteful. We continue to hear that these beings are built specifically to complete certain tasks down to the genetic level. Who are designing these entities if so? The next paragraph gets really technical, so bear with me here as I try with my non-existent PhD to explain. Speaking of genetic engineering, following sequencing of their genomes, we noticed a troubling and universal characteristic in the 5' of the regulatory sequence. Quick interruption, a bit of research tells me that the 5' end, which is usually at the beginning or head of a DNA strand, determines when and how much of a gene is, quote, turned on. The TPR are 134 base pairs, sequences containing, as its name suggests, 3 palindromes. In genetics, a palindrome is a DNA sequence that, when read in the same direction, gives the same sequence on both DNA strands, meaning the sequence reads the same forward on one strand as it does backward. They serve both as a flag and as a binding site for proteins. The 3 palindromes in the TPR are distinct from one another and have been poetically named 5' TPR, MTPR, and 3' TPR. The TPR is composed of 5' PTPR, 12 base pair spacer, chromosomal address, 12 BP spacer, MTPR, 12 BP spacer, gene address, 12 BP spacer, and 3' TPR. Phew. The chromosomal address is composed of 4 base pairs and is identical in each TPR of the same chromosome, but distinct between each of the 16 chromosomes of the genome. The gene address is a 64 base pair sequence that is unique for each gene in the whole genome. It is therefore understandable that the TPR serves as a unique address not only for numerically identifying a gene, but also for identifying its chromosomal location. For those with only a basic knowledge of genetics, this is completely unheard of. No living thing in our biosphere has this kind of precise address in its genome. Once again, the presence of TPR cannot be explained by evolutionary pressure, but only by genetic engineering on a genomic scale. Long story short, amazing precision seen at a molecular level. Again, if any molecular biologists are listening, let me know what you think of that section specifically.
I personally think the little aliens with the big black eyes, those are androids. They're biological robots. They're just programmed. They're no different than us cloning sheep. They just clone a being that can travel through space, because space
or
is very
some sort
big.
of an artificial intelligence thing that
It
doesn't
is
have
AI.
a life.
The rumor is it in the back of their head is a transmitter. So you got to wonder where it's sending shit.
TPR opens the door to several possibilities. One of them suggests that EBO geneticists can insert or remove a gene from a cell in a way that is far more targeted and efficient than our technology allows. No proteins have been identified in the EBO genome that interacts with TPR. Rather, we believe that these proteins are exclusively targeted by external genetic engineering tools, probably used at the zygotic stage of embryonic development, which is the earliest stage of an embryo. The nature of these tools is unclear, but we definitely don't have anything like them. The probable absence of these proteins from the genome is a further indication of their artificiality. Given the ease of modifying it with biomolecular tools, it's not out of the question that there could be polymorphism, which is the existence of distinct genetic variations or physical forms within a single population. In other words, an individual could be genetically designed to have characteristics that give it an advantage in performing a given task, like soldier ants and worker ants in an anthill. Note that these previous statements are speculation. To my knowledge, only one individual genome has been sequenced. I can't make a definitive statement on genetic variation between individuals. I've talked a lot about intergenetic regions. Now I'll briefly discuss intragenetic sequences, which are the structural regions found within a gene itself rather than between them. Briefly, because there's not a lot less to say despite obvious importance. Much like ours, their genes have silencers, enhancers, 5' UTRs, exons, introns, 3' UTRs, etc. There are many genes analogous to ours, which is not surprising given the compatibility of our cellular machinery. What's disturbing is that some genes correspond directly, nucleotide by nucleotide, with known human genes or even some animal genes. For these genes, there doesn't seem to be any artificial refinement, but rather a crude copying and pasting. Why they do it is nebulous and still subject to conjecture. There are also many genes which are not found in our biosphere whose role has not been identified. Finding the purpose of these novel genes is one of the aims of the program. I'd like to note before going any further that this heterogeneity of genes of known and unknown origin is undeniable proof of the artificiality of EBOs. To conclude with genetics, the mitochondrial genome, the separate independent DNA loop found inside the mitochondria, at the time I was working there had not been sequenced. It is safe to assume that this genome would also be streamlined and possibly has some version of TPR. To me, this is really something to think about and interesting. Why do each alien species look different? Different genetics, of course. We always hear about standard grays, but what about the famous Virginia alien, described with browner skin, protrusions on its head, and red eyes? What about reported taller grays? Maybe these beings were designed with different tasks in mind. Fascinating to think about. This is a good place to wrap it up for part 1 of this series. I am hard at work working on part 2. We covered the leaker's working environment and also the genetics of the supposed beings. The next section is very explosive, talking about physical descriptions of the supposed beings, potential tie-ins to how their craft work, cattle mutilation, and a lot more. Let me know down below what you think of this leak so far. Catch you next time.